Our final speaker in this first session is Dr. Amy Pasternak. Dr. Pasternak is a clinical assistant professor of pharmacy and clinical pharmacist in the clinical pharmacy department, and she'll be speaking about how she uses MGI data to understand pharmacogenetics research.
So good morning, everyone. I'm very excited to be here to sort of give some examples of how we're using MGI data to perform pharmacogenetic studies in our population from work that I'm working on and other collaborators at the College of Pharmacy have started working on. So for those who might not be as familiar, I just wanted to quickly review what pharmacogenetics is and sort of what we think about when we're looking at these types of evaluations. Pharmacogenetics is looking at how genetic variation impacts your response to a medication therapy, and that can be improved treatment efficacy, or it can be related to an increased risk of having an adverse event to that medication. And we typically think about modifying a drug response through two different types of mechanisms. The first of those is pharmacokinetic mechanisms, where you're thinking about how the drug is absorbed, distributed, or metabolized through the body. The most common that we typically see in clinically significant pharmacogenetic interactions is our drug metabolizing enzymes, or our CYP450s. And increasingly, this is becoming standard of care in clinical practice as we're prescribing medications. So this example is a new medication that was recently approved called siponimod for treating MS. And patients actually have to be genotyped for CYP2C9 prior to prescribing this medication because the genetic variants increase the risk of toxicity. And so if a patient carries a genetic variant, the drug's actually contraindicated in some patients. The other type of example that we see is pharmacodynamic drug-gene interactions. These are where you might have an increased sensitivity or response to a medication. The most clinically relevant example that I have seen is the approval of CFTR modulator therapies. And so only patients who have very specific mutations in the CFTR gene are candidates for these therapies because they're targeted to that type of mutation. So increasingly we're seeing these drug-gene interactions in practice, but most of the work that we are currently working on is really in this pharmacokinetic realm, particularly with these drug metabolizing enzymes. Because the majority of drugs are metabolized by a small subset of enzymes, there have already been extensive efforts by multiple groups and consortia to really define the functional consequences of the most common haplotypes in these genes. These groups, such as PharmD and CPIC, have developed a sort of standardized nomenclature that we use in pharmacogenetics called star alleles, which classify those haplotypes to make it easier to translate that to a clinical recommendation. And many of these medications actually have already been evaluated to such an extensive level that there's actually clinical guidelines developed by the group CPIC that provide recommendations of if and when you know a genetic result for a patient, what do you actually do with that drug therapy? We might know that the drug interacts with the gene, but what do we actually do with that patient's dose or with that prescribing decision for that patient? And so for some medications, we have that information available, but for many, that information is still missing. So that's where we have really been able to leverage the information that's available from the MGI cohort. There's really two types of studies that we've started developing in this cohort, and I'm going to go through some examples of both of these types of studies. The first that we've started working on are really discovery and validation studies of pharmacogenetic relationships in MGI. So because we know for a lot of medications what routes or enzymes they're being metabolized by, we can do a much more targeted analysis of our genetic information. We don't necessarily have to look at the whole genome, but we know we want to look at this specific protein, and we already have categorized what the most common and functional consequences of those haplotypes are. So first we picked a medication that we were interested in evaluating, and for my postdoc work and residency work, that drug happened to be tacrolimus, which is an immunosuppressant that is used in a lot of different types of transplant areas. This medication is preferentially metabolized by the CYP3A family of enzymes, but interestingly, it's preferentially metabolized by CYP3A5, which is an enzyme that the majority of individuals actually do not express because of a common no-function variant in this gene. The concern with this pharmacogenetic relationship is that the drug is very sensitive to the exposure. If your drug exposure is too high, you're at a very high risk of toxicity. And if the drug exposure is too low, you're at a high risk of having a graft rejection from that transplant. And patients who are expressors of this enzyme are at a higher risk of having lower exposure and therefore a higher risk of rejecting their graft and require much higher doses of this medication. So this is an example of a medication where there is already a lot of evidence to say when we know a patient's a CYP3A5 expressor, we need to potentially double the initial dose of tacrolimus to get a therapeutic drug concentration and maximize that efficacy and safety of the medication. However, this medication is administered via multiple routes, and the majority of the existing literature is really focused on the oral administration of the medication. However, a lot of patients, particularly who are receiving bone marrow transplant, receive intravenous tacrolimus because of the side effects of their conditioning regimens. And so there's really a lack of data of what do we do with these IV patients. The way the guideline is currently written, there's no differentiation between route of administration. But what we've really hypothesized is that this pharmacogenetic relationship will likely differ between these two routes. And the reason for that is because the CYP3A family of enzymes is expressed in both the intestine and the liver. So when you give a drug orally, it's impacting both the absorption of the drug in the intestine and the elimination of the drug in the liver. However, when you give the drug intravenously, you're bypassing that absorption effect in the intestine, and it's really only being impacted in the liver. And so there's some small studies of up to maybe 100 patients that have identified our hypothesis or support this hypothesis that the relationship is different, but it's not a large enough cohort to really to begin to say, if a patient's getting the IV administration, what should our dose recommendation be, sort of like we have with the oral administration. And so the first step of sort of this process was evaluating what type of genetic information could MGI provide to us that we're interested in. We knew what gene we wanted to look at, and the consortia that have already assessed these genes have really well-defined haplotypes and frequencies in populations for this gene. And so the two most common variants that we would expect to find in our population based on the demographics of the University of Michigan cohort is the CYP3A5 and CYP3A*6, CYP3A5*3 and CYP3A5*6 alleles, which are both no-function variants. And when patients are homozygous or compound heterozygous for these variants, they're CYP3A5 non-expressors, and that's really the phenotype that we're interested in. If a patient carries a wild-type allele, then they're a CYP3A5 expressor. And so we were able to identify that MGI would be able to give us the clinical data that we would need to categorize our patients into our phenotype groups of interest. So with that information, we initiated a retrospective review of bone marrow transplant patients who were receiving IV tacrolimus. It's great that we're able to leverage all of the other clinical resources that are available to researchers at Michigan Medicine, where we can link the MGI data to DataDirect data to identify our cohort. We can link that back through Immerse to look at more granular adverse outcome data that might not be captured in DataDirect, as well as link that back to internal transplant databases that have survival and graft outcome information. And so this was a really nice workflow for us to identify our cohort with those resources, then be able to submit a targeted variant request to MGI for our cohort of interest so that we could categorize the patients that we were interested in evaluating. So data collection for this project is still ongoing. We anticipate we'll begin our analysis early in 2020. And ultimately, our analysis will be evaluating, does the CYP3A5 phenotype impact, one, our IV tacrolimus exposure, and also we want to look at, does it impact our dose conversion requirement? All patients are eventually converted to oral therapy, and right now every single patient is converted at the same ratio. But likely there might be some personalization opportunities there to ensure patients maintain therapeutic concentrations through that process. And we feel like the impact of this will be, one, defining, does phenotype need to be considered in our dosing decisions? It's possible that it doesn't, but if it does, we finally have a large enough cohort where we can really start to begin assessing what type of dose recommendations do we need to be making in this population, which is really unique. In addition to sort of these single drug specific gene types of analyses, of which there's lots of examples of what we've worked on, there's also unique opportunities in MGI to look at how pharmacogenetics impacts patients on a larger level. Over 58% of the MGI cohort has received a medication with a clinically significant drug-gene interaction, and that's defined by either having an FDA package labeling recommendation to modify therapy based on a genetic result or a clinical guideline to modify therapy based on a genetic result. And so lots of the studies that have been conducted in large populations haven't had the linked clinical and genetic information like we have here in MGI. And so there's sort of these unanswered questions of how frequently are drug-gene interactions actually occurring in our population, what's the overall impact of that to the patient, to the health system, to the payer. So there's lots of opportunities for additional analysis in this area to look at the population impact of these drug-gene interactions. So there's lots of other examples of discovery and validation studies that we can perform, but I also want to provide a quick example of the translational study opportunities that we've identified that are available here in MGI. And Dr. Hertz briefly mentioned this in his prior question. So there's opportunities to identify, because we know drugs and genes have interactions, but not all of this genetic testing is currently standard of care, there might be opportunities to identify patients who are at risk for toxicity based on MGI data, and how can we translate that into clinical practice decisions. One specific example of this that we're currently investigating is for the gene dihydropyrimidine dehydrogenase, or DPYD, which is the primary enzyme that's responsible for metabolizing fluorouracil or capecitabine, which are two commonly used chemotherapy agents. It's been really well defined in the literature that patients who have DPYD deficiency because of these genetic variations have a significantly increased risk of severe toxicity and an increased risk of fatality due to this toxicity. But because these variants are very rare in the population, testing for this gene hasn't become standard of care in general practice. But the most common DPYD genetic variants that are associated with toxicity to these medications can be identified in MGI. And so there's the opportunity to identify patients who are going to get these medications who are at increased risk of toxicity based on their MGI genetic result, and make an intervention of performing that confirmatory clinical testing so that that can be used in the treatment decision of the prescribing oncologist. And so this study is being developed and led by Dan Hertz. It'll be a proof of concept pilot study really to determine what's our feasibility of identifying these patients in time and performing and confirming this clinical genotype result. And then based on the ability to confirm that MGI genotypes are accurately predicting patients at risk of toxicity, how could we scale this to look at other pharmacogenetic markers to do a much more precision confirmatory genotyping approach in these populations? So I just want to quickly thank everyone who's been involved in these studies. I'm sure I probably missed someone. It's been a great effort. And then I'm receiving support from AACP for the IV tacrolimus study. And with that, I'd be happy to take any questions.
So I noticed that you're focusing on IV infusions and things that are given in the hospital. Have you looked at outpatient prescribing, and do you have any advice for people looking at that?
Yeah,
and how accurate that is?
Yeah, so we're still figuring that out. We are looking at outpatient for the oral dosing to look at some of those dose conversion requirements. The strategy we have used is for every single patient, we are actually going into a MRS to confirm the outpatient dosing at the time, because the data direct export is not always accurate with outpatient dosing if they don't change the prescription in the medical record. So it's been a highly manual process at this point to look at that.
This is actually a comment on that. The MGI is now linked to SureScripts. So at least in principle for MGI participants, even if they get the prescription in a different medical system, we ought to be able to pick that up.
Yeah, I agree. Part of the challenge is a lot of times if they're adjusting doses by a 0.5 milligram, the prescription still just says a 0.5 milligram of tacrolimus, but then in the clinical note, the physician will adjust the dose.
Yeah, you would not have access to that.
Correct. You would not pick up on that in that process.
Thank you.
All right. Thank you.
All right, well, thank you.